Good afternoon. Thank you very much for the opportunity to speak here. Thank you for taking the time to come here after lunch. I'll tell you a little bit about some of our work on piecing together different descriptions of the brain. Our jumping off point is this idea that most of us in the room are engaged in one way or another in the enterprise of generating brain maps. So you can do this when you're doing task, fMRI, cortical thinning, and patients versus controls, or any of the other very exciting techniques that were shown over the past couple of days. But when you have a map in front of you, it naturally draws you to wonder, what does this map look like? Does it co-localize with other canonical features of brain structure and function? And what I mean by that is, does it look like patterns of gene expression, neurotransmitter receptors, and so on? The reason you want to know this is you want to have some idea of what are the underlying mechanisms that gave rise to that map, but then also what should be some of your follow-up experiments. And it's not as though this type of data is completely inaccessible to us. As I hope to show you over the next 20, 25 minutes, we can actually get a lot of these descriptions either via techniques that we're already very familiar with, or data sets that are shared openly across the globe. So where is all this data? Well, I'll show you a couple of things that we've done over the last few years. This is a toolbox called Abigen that I can't not show. It's our tool for working with imaging transcriptomics. So with just one line of code, you can download and pre-process the bulk sequencing, the single cell sequencing, and the Allen Atlas works with a human, mouse, et cetera. And then once you have that data, you can generate maps for genes of interest. You can superimpose any kind of regional mask to get a complete data set for your ROI. You can use it to generate densely interpolated maps and so on. Once we started to get more interested in how different levels of organization are related, we came up with another tool that was shown a couple of times already, NeuroMaps. It's kind of like a Google Maps for the brain. And the idea is that basically if you come in with an input map and you want to try to interpret it or contextualize it, we provide you with a suite of transformations to go to and from different coordinate systems that we have in brain imaging. We have a curated library of various biological features and then statistical tests for doing spatial correlations. That's what some of this library looks like. I think it was, again, already discussed a variety of things, both from BigBrain, gene expression, and so on. The one component of this that we had to do quite a bit more legwork for are the neurotransmitter receptors. So we cold called a whole bunch of PET researchers around the world to ask them to share their data, and at the end what we had was a whole brain atlas of 20 different receptors and transporters. I also want to just very briefly mention, because people might not know this, but we have a fully functioning prototype for NeuroMaps in the mouse. And this is really exciting because we now have a pretty good handle on how coordinate systems work there and how the hierarchy of different regions should be organized. We have lots more data sets, things that we don't necessarily have in the human. We have ways of actually plotting things as well, and it's compatible with all of the functions that we've put out in the past. The thing that I want to talk about for the rest of this talk is what kind of new questions and scientific directions does this type of data unlock for us? Kind of the prototypical two examples, at least in my mind, are one, multi-omic assays, and two, benchmarking. So what I mean by multi-omic assays is exactly what I just showed. You come in with an input map that you like, and then you compare it to everything that we have in the library, kind of like an NMR, spectroscopy kind of spectrum, and then you figure out exactly what are the other biological features that your input map is related to. And so we've done quite a bit of this work relating microarchitecture to dynamics, microarchitecture to disease, and so on. I thought I'd switch things up and just for variety, show you something that's not in the human. So this is just an example of our work in the mouse. So the fundamental question here was, we tend to think of different architectural features shaping dynamics and activity, but really the one element that's really important are the synapse types. And what you might not know is that there are many different synapse types, in fact, probably hundreds, depending on what scaffolding proteins they express, their morphology, and so on. And each of them is capable of presumably translating that chemical message that comes in a slightly different way and generating different types of dynamics. So what we have here is a really cool data set of whole brain single punctum extractions, so about 1 billion synapses, and we have whole brain maps of 37 different types of synapses. And what we're going to do is compare them to awake fMRI recordings from those same areas, again, across the whole mouse brain. We're going to use a technique where we basically do extensive time series phenotyping, so we generate many different time series features for each brain area. And we ask what types of synapses co-localize with different features of dynamics. And what's really cool is, so this might not mean much to a lot of you, but there are these two very important kind of broad categories of synapses, those that express PSD95 and those that express SAP102. So these are two types of scaffolding proteins. And one of them really is spatially correlated with regional differences in stationary dynamics. So it ensures that you have nice, coherent, predictable ongoing dynamics with nice autocorrelation. The other one helps you to do exactly the other thing. It's generally associated with intermittent high amplitude events. So it's really nice, I think, when you have data like this to look at some of these elemental questions of how structure shapes function. The other application after that is that, but one that people don't consider as much, is that once you have a lot of these data sets under one roof, you can also do extensive benchmarking. So you can ask, for instance, how consistent are different technologies, how they can be used to augment each other, and so on. And so one example would be, for instance, if you have two techniques that ostensibly measure the exact same thing. So you have autoradiography and PET imaging that are measuring neurotransmitter receptor distributions. Or if you have two techniques that are measuring 2 points in one very circumscribed, very strongly interrelated pathway. So for instance, gene expression and a PET mapping of protein density that could be separated by alternative splicing and protein buffering and so on, but should be correlated with one another. And then you can obviously use these techniques also to augment each other, and sometimes, as you know probably, autoradiography is used to augment PET imaging. So this is just an example from this. It's actually from the famous Jülich autoradiography data set, where we're looking at how PET estimates of receptor density are correlated with autoradiographic measurements of those same receptors, and we're doing this across different layers of the cortex. The other thing that you can do, and I think is very exciting, is you can move beyond the maps and actually try to think about how they relate to the underlying neural wiring patterns. So traditionally in network neuroscience and connectomics, in order to understand how different areas are communicating with one another and so forth, we tend to abstract away a lot of biological detail. We tend to represent them as graphs where all the nodes are the same. But we know that's not true. All brain areas are different, and they're different across a variety of features, some of which I just spoke about. So what you can do very simply is take some of the maps that I just showed you. You can superimpose them on your favorite wiring diagram from diffusion MRI, track tracing, and so on. You can assign values from that map to the different nodes and then generate these beautiful objects that we call biologically annotated connectomes. In network science proper, they might be called colored graphs, but what they are network architectures where the nodes now have either scalar vectors or even multidimensional scalar values or even multidimensional vectors attached to them that tell you something about the local neuronal populations. So you can play this game for any of the features that I just showed you. And what's really cool about this is now you can ask a variety of fundamentally new questions about how network architecture interacts with the local biology. So for instance, on the left-hand side, you can ask if two areas that have similar local features are more likely to be connected with one another, whether they show evidence of assortative mixing. You can ask if you have nodes that are putative hubs, have lots of connections, does that entail additional metabolic cost, or are they somehow privileged in terms of their dendritic branching, et cetera, et cetera. You can also use these to make better models. So you can use annotated connectomes to make better models of brain function. You actually saw some of those examples already. You can use them to make generative models of how the brain develops. And finally, and I think you also saw this again from Alan's talk, looking at, you can use these annotated networks to make better models of disease spread as well. The really cool thing is that you can also use annotations to move beyond this particular paradigm and generate entirely new types of networks. This is not entirely foreign to us. In fact, we do this all the time. So for instance, if you have recordings of neural activity in two brain areas, you can correlate their time courses and you get a functional connectivity matrix. Not too mysterious. Even before we were doing that, folks such as Alan were taking pairs of areas across many different individuals, correlating, say, their cortical thicknesses to generate anatomical covariance networks. Or if you have multidimensional information and each node, you can just correlate, but that doesn't have temporal dependencies, such as many genes. You can correlate those signatures and generate things like correlated gene expression networks. So once we had all of these different annotations, we decided to ask how these different levels of organization relate to one another. So we have region by region biological interrelationships in terms of gene expression similarity, receptor similarity, laminar similarity, similarity in metabolic time courses, hemodynamics, CFIS, and so on. And I'm not going to show, this is actually a cool paper, but I'm just going to show you a few things. One is, if you look at the matrices or even the 3D diagrams, you can see that although there is a lot of structure in these networks, they're actually very different from one another. And even if you ask seemingly very simple questions, such as what are the hubs of these networks, what are the dominant gradients, you can get very, very different answers. The point being that each of these levels of description is telling you something slightly different about the propensity of areas to develop together, to be modulated by ascending projections, to be affected by external input, and so on. And looking at any one of them in isolation is really not enough to understand how all of them, what we really should be looking at is how all of them come together and what complementary information they provide. So that's all well and good. We have the data, we have some of the methods. Is it really that simple? Can we just use the same stats that we were using up until now? And the answer is probably not. So we lose a lot of sleep thinking about this. We've written a few papers trying to explain to people, what would be the best way to deal with these new data sets and what people should do or maybe shouldn't do. So we've done quite a few benchmarking studies looking at how one should control for spatial autocorrelation when relating maps to one another. You saw also that in Jessica's talk yesterday. This is something that I'm really proud of. We recently came up with a method that allows you to randomize networks that actually have biological weights on them. As far as I know, it's the only method that we have in the field, and it works pretty much perfectly. And then the last thing is we are actively looking at what are the best methods to perform spatial interpolation when you have sparsely sampled data. This, I think, is going to be a big challenge for us as a field going forward, because obviously with gene expression, we have samples being taken not everywhere in the brain, but in a small set of areas. This is true of intracranial EEG and so on. And I think understanding exactly how different methods work with the kinds of coordinate systems that we have and the types of data sets that we have is very important. Once you have also many different maps of brain organization, you can ask actually how systems that should be related, how they work together. So I want to tell you a little bit about that. I told you a little bit about neurotransmitter receptors, but there is another class of chemical messengers in the brain that is more ancient and in some ways more fundamental, and those are neuropeptides. So neuropeptides, for those who don't know, they're larger amino acids. They also get released from the presynaptic boutons, but they get released to the side, so their action is a little bit more diffusive. And they also, just like metabotropic receptors, they work via G-protein coupled mechanisms. So, and the cool thing is they had functions in the body, but their function has changed over time, and they also have different functions in the brain. So, using data from the Human Protein Atlas and the Allen Atlas, we recently constructed this data set. It's another atlas of 38 different neuropeptide receptors across 14 different neuropeptide families. Again, just because I'm showing the cortex only, don't mind that. It's actually the whole brain. The atlas is really cool, and I hope to convince you that over the next couple of slides. So these are the expression of these different receptors across different anatomical structures. So we find these really cool peaks of expression, for instance, oxytocin receptors in amygdala, peak opioid receptors in the accumbens, leptin and hypothalamus, and so on. There's very dominant cortex to subcortex gradient that you see, I think you can see there. And then this is kind of just an interesting aside. So sometimes when you see like what the actual samples from the Allen Atlas look like, where they're taken from, you can be fooled into thinking that they were taking very random locations. But actually they had a lot of foresight. And when they sampled certain structures, they sampled them very, very well. The hypothalamus, for instance, which secretes many of these neuropeptides, they sampled each of the 8 nuclei of the hypothalamus. So that's what you're seeing here. And what's really interesting is that there's this really dominant axis that goes from the tuberal hypothalamus, so ventromedial, dorsomedial hypothalamus, and so on. It goes from there to the mammillary complex, which is more posterior. And the reason this is interesting is that these nuclei, it really nicely recapitulates the stages of embryonic development of these nuclei. So the point that I actually wanted to make here is that sometimes we go into really, whole brain maps, but sometimes you can really zoom in into finer structures, and I think there's a lot of value there as well. Talking about how annotations can help us to make better models of dynamics, here what we did was to say, okay, well, does neuropeptide signaling help me to predict functional connectivity? So we take our neuropeptide receptors, we do an outer product with their cognate ligands, so that we have this matrix of potentially how much binding there is between a receptor and a ligand. And then we mask this with a white matter connectome. So we generate a whole brain white matter connectome that's weighted by the amount of neuropeptide signaling that takes place along the edges. And then we try to predict either hemodynamic connectivity from fMRI or electrophysiological connectivity from MEG. And so that's what you're looking at here on the y-axis. You can see how well we predict fMRI functional connectivity. MEG's functional connectivity is on the x-axis. You can see a tiny little point right there that says no annotation. So that's just how a connectome with no neuropeptide weights would do. But then you see really cool things where basically a lot of these different receptors actually help you to predict different types of data. So for example, natriuretic peptides, which are known to modulate blood pressure, predict hemodynamic connectivity. Somatostatin, Y-amides have to do a little, also relate, very well known to be related to oscillations, electrical activity. They tend to predict MEG connectivity. And then endothelins, tensins, and the like, so things that modulate the tone of blood vessels, they help you to predict both MEG and fMRI. So the point that I want to make here is that by really thinking about specific forms of communication, we can get into that, what is a very huge gap between structural connectivity and functional connectivity and build more biologically realistic models. The last thing that I want to show you on this is to ask how these chemical signaling systems are related to one another. So as I said, they tend to get released in the same place, and in fact, neuropeptides also work via G-protein coupled receptors. So, and there's lots of evidence of co-transmission of the two. So that's what you can see here, because we have both the neurotransmitter atlas and the neuropeptide atlas. We can actually compare them to one another. So in this like little Venn diagram, what you're meant to get out of that is that there's the spatial distribution of neuropeptides and neuroreceptors actually share about 60% of their variance. So they are actually co-localizing. And what's really cool is in the bar plot, you see this actually being mostly driven by metabotropic receptors, which also obviously by definition use G-protein coupled mechanisms. So that's nice. Then the last thing that I wanted to show you here is we can actually ask how these signaling systems evolved over time and co-evolved. So this is maybe things that people haven't encountered before, but what we're doing here is we're taking the amino acid sequence of each one of these receptors, and we're looking at how much it changed at different points in our phylogeny. And what we're specifically looking at is the rate of change of the amino acid sequence at different points. So that's what's being shown here, the substitution rates on the left-hand side. So this is a measure of how much positive selection there is at any given point in time. And what you can see is that this positive selection pressure really peaks for all three around the transition from amniotes to mammal tetrapods. But the really cool thing is what's shown on the right. If you just take the medians, you can see that contrary to what you would see in a textbook, the inotropic receptors are almost immediately locked in, and they don't change much at that point. But the evolutionary changes, the evolutionary pressure for slower signaling, so for metabotropic receptors and neuropeptides, is much more protracted, meaning that they continue to acquire new functions over time. And for anyone who's interested in what these functions are in the brain, I invite you to take a look at the preprint there. The point that I also wanted to make here was sometimes you don't even need to, you can use the brain data as a jumping off point to look at relationships between systems on some other level. Okay, the other thing that I wanted to talk to you about is our support systems. So again, a lot of us are talking about structure, function, and we tend to think of architecture, dynamics, things like that. But all of this costs metabolically. It costs in other types of resources. And blood supply is something that's very fundamental to this whole thing. Camille talked about it, but we sometimes tend to not consider it as closely as we should. So we recently did a project where we took ASL data from HCP development and HCP aging that have really nice sequences. And if you stitch them together, you have this kind of like nice lifespan data set from about 5 years of age to about 100 years of age. And I'm just going to show you here, this is the normative map of regional differences in perfusion. I just wanted you to take a minute and just gaze upon how beautiful this is. So it's on the flat map, we're overlaying the Glasser parcellation. And the Glasser parcellation, as you probably all know, is not derived from ASL. It's derived from T1, T2, resting state fMRI, and so on. But I think you can appreciate just how much just how much of the areal differences align with just the underlying differences in perfusion. So perfusion is really clearly very, very important to the differentiation and arealization of the cortex. Again, in the spirit of kind of querying how different systems are related, in the neuropeptide data that I just showed you, we actually have two receptors that are known to modulate vasoconstriction and vasodilation, endothelin A and endothelin receptor B. And so one is kind of like a low-pass brake, the other one a high-pass brake for blood flow. And so they should be related to perfusion, but they should be related in completely different ways to perfusion. And that's what you can see in the scatter plots. We're plotting regional differences in endothelin receptor distribution, and then against regional differences in perfusion. And you can see one has a really nice positive correlation, the other one has a really nice negative correlation. Again, having all of these data sets in under one roof can really open up, in my mind, very exciting, very important questions. The last thing that I want to show you here is you can also actually take this perfusion data and you can compute gradients from it. And if you do that, you can actually really nicely recover all the different arterial territories that normally you'd have to go into a stroke data set to find. So they come out in a completely data-driven way for free. You can also identify border zones, as we show in the paper. And then putting that everything into a semantic space, that's the graph on the bottom right-hand corner, and overlaying everything with Neurosynth, you can, see exactly how much of different cognitive functions are being supplied by different sources, sources of blood supply. And also, you can predict very well what the symptoms would be if an occlusion or a stroke were to occur in any one of those territories. Okay, so everything that I've set up until now revolved around this idea, okay, we're going to take the brain, and we're going to just drill deeper and get more and more info for all areas. But You can also think about extending the canvas and going into bigger maps. So this is an example from a recent study where we, kind of working with Marta Bianciardi in Harvard, we delineated 58 different brainstem nuclei and then reconstructed their projection maps to the cortex. And so what you have now is not necessarily a kind of deeper representation of the cortex, but just a bigger network. And again, it leads to really, I think, fundamental insights, and kind of a very consistent finding that in that paper is that so much of what we think we're seeing purely in the cortex actually comes from the brainstem. So this is, for instance, a map of how different brainstem nuclei connect to the cortex overall, and it's really nicely correlated with the dominant rhythm in the human encephalogram. The alpha rhythm. If you look at how similar areas are in terms of their brainstem connectivity, you get the famous unimodal transmodal gradient. And the reason this is interesting is that, from the work of Daniel Margulis and Marcel Meslem, like the implication is that this is a really compact cortex-only phenomenon, this differentiation of unimodal to association cortex. But here you see that it actually has clearly a subcortical origin to it as well. So again, making bigger maps with more detail, potentially very cool. And then the last thing that I'll show you is, people who collect data, they're doing all the work, like 99.9% of the work. And then there's us who are like the middleman, and we do like a little bit. But what if you could do no work whatsoever, and you just use the literature instead? So what we did here was to take transformer experts, so LLMs. We took a whole bunch of off-the-shelf LLMs, so ChatGPT, Gemini, and so on. Actually, we also have Claude, which I didn't list there. So, 6 LLMs, and we asked them, okay, for a particular region, what do you think is the probability that it's involved in function X? And so you get a whole bunch of maps for different functions, which I'll show in a second. The obvious thing to do here is to compare it to something like Neurosynth. And you can see that generally we get very good concordance. But one thing that Neurosynth can't do, because it's looking for peak MNI coordinates in fMRI studies, is it can't give you anything else. Whereas with the LLMs, we can, for instance, get maps for different model organisms. And this is just to show you how good they actually are. This is on the y-axis, you're seeing maps from this famous International Brain Observatory data set that just got released. So these are neuropixel recordings across thousands of mice. They're all doing the same task, mind you. We have their annotations. And on the x-axis, you're seeing the consensus maps from our LLMs. And what you can see is that when they present the stimulus, we have the peak correlation with our vision map. When they do choice velocity speed, so the mouse is responding, we have peak correlations with manipulation. And when they get the feedback, which is auditory, we get peak correlations with listening. So it can clearly work with other species. It can also discover latent features that, other than just the maps that it's producing. So one way to kind of get these unimodal transmodal gradients is to look at T1, T2 maps, so like intracortical myelin. So we can do this. That's what you're seeing on the y-axis in the human, macaque, and mouse. And then we can try to see if we can find the same gradients looking at the meta-analytic results from LLMs. And again, you can see that they can all discover additional features that are sort of more hidden in the data. And the last thing I'll show you is that they can also identify causal circuits. So in the middle there, you see a data set. These are people who had intracranial electrical stimulation and in different parts of the brain, and they were asked to report their subjective experience following stimulation. So it's like a big data set of like 70 people or something like this. On the left-hand side are the correlations, the spatial correlations with Neurosynth. You can see that they're very poor. And on the right-hand side are the correlations with the LLMs, which you can see are very high. And again, the LLMs are not using just fMRI, they're using everything. Every molecular paper, every fMRI paper, every PET paper, every anatomy textbook. And so, they seem to work pretty well. Okay, I'm going to end there. Just to say, I hope I've convinced you that neuroscience is increasingly a data science. We have lots of exciting tools for representing data, for working with data, and we're, I think, going towards an increasingly vertically integrated understanding of how brain structure and function are related. And this has brought with it new questions and I think a fundamentally different culture of how we do this. So I'll just say that if you're interested in anything that I just said, we recently wrote a couple of papers on this topic, and so I invite you to check that out. Thank you for your attention.